right, welcome to the second segment of chapter 14. We're going to talk about things that are a little bit more complex than what we were talking about before. So Mendel's P's showed relatively simple inheritance patterns. Now we're going to talk about some more complex inheritance patterns. So inheritance patterns are often a lot more complex than what we were just discussing. They could involve more than one gene, more than one allele, some complex, other traits that are associated with that particular character. So let's talk about some of these. So some examples, as I pretty previously mentioned, we were dealing with the single gene characters. However, they could deviate from the simple Mendelian genetics that we were just talking about. You could have a situation where one allele is not completely dominant or recessive over or under another allele. Now, some genes could have more than two alleles. So we were talking about only one of two alleles, right? There could be more than two alleles for a particular gene. And then, as I mentioned, one gene could produce multiple phenotypes that might be associated with that. And these are often seen with, disorders and diseases where you see a lot of symptoms associated with that particular disorder. So let's talk about degrees of dominance first. So there are different or varying degrees of dominance. We've been talking about true dominance, where it's basically called complete dominance. It occurs when the phenotypes of the heterozygote, right? So we can see here we have the capital P and little p, heterozygote, and the dominant homozygote. So that's a homozygous individual with both the dominant alleles. Both of them are going to have the same phenotype. And in the case of the flowers, we can see that the homozygous dominant and the heterozygotes are all purple, and it's because the allele for purple is dominant over the allele to white. However, this doesn't always occur this way. Sometimes there's what's called incomplete dominance. So this is where the phenotype of basically a cross between two opposite phenotypes and genotypes, right? Two true breeding. And in the case here, we have red flowers plus white flowers result in heterozygous individuals, which will be pink. Now, this is like a blending of red and white, right? So we can kind of see that red plus white, blending them would create pink flowers. Well, there's also what's called codominance. Codominance is where there's two dominant alleles, or they're about equally dominant, and it affects the phenotype in separate ways. So here, going back to the flowers where we would combine the red with the white, we could see red plus white would result in both both of the colors, right? So you would see red and white. Now this may not make sense until we see this picture right here. So here we could see the dominant and recessive. So the dominant ones would be red and the recessive would be white if they had two of the recessive alleles, and that would be the case of complete dominance. Here, this would show the flowers under incomplete dominance regarding color, and then this would show the codominance. You can see that you can see both the red and the white in a single flower. All right, so when we do the cross, this is how they're notated. Okay, so here we're talking about flower color, so we use the capital C, and then the superscript represents the actual allele. Okay, so for the red ones, we have C, and then superscript R, and then for the white, we have C, superscript W, and these are both homozygous, opposite genotypes, so they're true breeding for each one. When you cross them, you end up with all pink. Okay, so it's similar to the other one, except all of them are going to have the heterozygote. Well, they're all going to have the heterozygote genotype. And then, of course, since this is incomplete dominance, we're dealing with heterozygotes that are going to appear pink. And then we cross the F1 generation to get the F2 generation. We get, instead of a 3-to-1 ratio of colors, we get a 1 to 2 to 1 ratio of colors. We get 1 red, 2 pink, and one white. So it's the same for the genotypes as well. Okay, so we have one homozygous dominant, two heterozygotes, and one homozygous recessive. So we have both a 1 to 2 to 1 ratio of genotypes and phenotypes. Okay, so let's talk about the relationship between dominance and the phenotype, and of course, recessiveness. It's important to note that the dominant allele does not subdue the recessive allele, and it doesn't mean just because it's called a dominant allele means that it's more superior to the recessive allele. It's not usually the case, and really superiority or inferiority really has nothing to do with it. Alleles are just variations of the nucleotide sequence of a gene, so it's just different, and they're controlled in different ways. Now, for a particular character, dominance and recessiveness, and the relationship between dominance and recessiveness of those particular alleles, it can depend on the level in which we look at the phenotype. Here's an example. Tay-Sachs disease. It's fatal, and it's the result of a dysfunctional enzyme. This dysfunctional enzyme results in the accumulation or increase in lipids in the brain. So at the organismal level, so we're talking about the organism, okay, the allele's recessive. So what this means that for that organism to show this particular disease, right, Tay-Sachs disease, that individual must have both of the recessive alleles on each chromosome. At the biochemical level, so here we're talking about the enzyme, okay, so the activity of the enzyme. The phenotype is incompletely dominant. Okay, so what this means is that, let's say we have a heterozygote individual. So this individual has one normal enzyme and one dysfunctional enzyme. Well, the activity of the enzyme is a blending of the two. So let's say the normal has a high activity, the dysfunctional has a low activity, but when you combine them, there's a moderate activity. Okay, so that would be in reference to the incompletely dominant activity of the enzyme itself. At the molecular level, the alleles are codominant. So what this means is that both of them will be expressed. Okay, so let's say we have a heterozygote individual, right? So that individual has both alleles, right? So one dominant, one recessive, but both of them are expressed. And that kind of can be related to what we see at the biochemical level, where you see both of the different enzymes, one's functional, one's dysfunctional, but its activity is incompletely dominant. Okay, so again, it can be dominant or recessive or codominant or incompletely dominant at varying levels of hierarchy. Now, another thing that's important to discuss is that just because an allele is dominant doesn't mean that it is in a high frequency in the population. Let's look at the trait polydactyly. Okay, so what polydactyly is basically more than 5 fingers and toes, or toes. This is dominant. However, only 0.25% of the population has this particular trait. So basically, everyone has the recessive condition, right? So everyone has the recessive, both of their chromosomes have the recessive allele. So this puts 99.75% of the population that have the recessive phenotype, which is 5 fingers and toes. So this occurs at about a frequency of 1 out of 400 births in the United States. Now let's talk about multiple alleles. There are some loci that have more than two alleles at each one. Now, most genes have two forms, right? So there's two alleles for each whatever trait. However, there are some key examples that have more than two alleles. So one of them is the ABO blood typing, or the blood groups in humans. There's actually three alleles. Okay, so they are denoted with an I with superscript A and B, and then the lowercase I, which would be the recessive Now, this actually displays something else as well, which we'll talk about. Let's talk about this. There's an enzyme that's encoded by the A allele. Okay, this adds what's called an A carbohydrate. Then we have an enzyme that's coded by the B allele, which adds the B carbohydrate. And then the enzyme that codes the I allele, which really adds neither the A or B, but it does add a base carbohydrate, and it's different from the A and B. Now, you can see here that the little letter I, okay, so this is for the O blood type, this is recessive. However, we have A and B are both capitalized, so this means both of those are dominant. So both A and B are dominant over the O. This is an example of codominance as well. So this is actually an example of multiple alleles and codominance. Now let's look at these carbohydrates, or how they're represented. So here we've got the alleles. We have the I superscript A, we have the I superscript B, and then we have the little I. So, like I said, the A allele is going to give an A carbohydrate, and this is shown by the triangle. The B allele is going to give the B carbohydrate, and this is shown by a circle. And then the lowercase I will give no extra carbohydrates on the surface of the blood cell. And I say extra because ultimately, on the surface of the blood cell, there is going to be an O-type carbohydrate. It's just basically present in the A and B individuals. Now, this kind of represents that in terms of genotypes. So these are the alleles. Now we have a bunch of different genotypes that can be associated with this. So those with A-type blood can be homozygous dominant or a heterozygote. Those with the B-type allele can have homozygous dominant or the B or the heterozygote as well. Those with AB blood type, like me, have both the A and the B, but not the O type carbohydrate. Basically, just the A and B are expressed on the surface of the blood cells. And then those individuals with type O blood have both recessive alleles. So this is kind of shown here. For the A type, it's only going to have the triangles on which represents the A type carbohydrate on the surface of blood cells. And those with B-type blood are going to have just the circles on the surface of the red blood cells. And those with AB have both. And those with O do not have any extra carbohydrates on the surface of their blood cells. And this shows the different blood groups, right? So we have A, B, AB, and O. And there's another thing that's associated with the blood type as well. That's the Rh factor. So you can be either Rh positive or Rh negative. And I will discuss that in a couple examples, which I will show you in a whole nother segment. Now let's talk about pleiotropy. Most genes have multiple phenotypic effects. This is called pleiotropy. Okay, so basically we have a gene, it's expressed, and the expression of that gene is going to result in multiple phenotypes. And not just, we're not talking about in as separate individuals, within the same individual. So for example, the alleles which are pleiotropic, so they're gonna be alleles that actually show these multiple phenotypic effects. They can result in multiple symptoms, which are seen in hereditary diseases like cystic fibrosis and sickle cell disease. So both cystic fibrosis and sickle cell disease result from a mutation in a single gene. However, there are a lot of symptoms associated with those two diseases. And these aren't the only two, but these are just some key examples. Now, some traits can be the result of combinations of more than one gene. Okay, so this could be, multiple genes coming together to kind of give a different form, right? Which is the result of the combination of all those genes. These are called polygenic. So an example would be hair color, eye color, and skin color. All of these are the result of different combinations of alleles from more than two genes, or more than one gene, often up to three genes. Because polygenic inheritance is the result of multiple genes, we could call them quantitative characters because they vary within a population along a continuum. Now, quantitative variation usually indicates that polygenic inheritance is at play. And remember, polygenic inheritance involves more than one gene. Now, skin color, as I mentioned already, is an example. So let's take a look at representing the three different alleles that contribute to skin color. Now, this could be the result of three alleles from, three different genes, right? So here we have basically these A, B, and C, and different combinations of alleles within each of those three, let's say, genes, result in the huge variation in skin color that we see in our population. Okay, epistasis is really cool. Epistasis is basically is the result of a gene at one particular locus, which alters or affects the expression of a gene in another completely different locus. An example would be the coat color in Labrador retrievers. Okay, so there are three possible coat colors. There's black, there's brown, and then there's the golden retrievers. That's the result of epistasis between two different genes, which are related to the pigment in the coat. So one of the genes determines the actual color. Okay, so it's actually going to be just black or brown. However, there's another gene that affects whether or determines whether that pigment gets deposited into the coat. If it doesn't get deposited, that's where you end up with the gold. This Punnett square, which kind of shows this process. So we can see we have a cross between two black labs, which are heterozygotes at both of the genes. Okay, so they're going to show the dominant traits. So here, the dominant trait here is black, and brown is recessive. And then we have the gene that affects whether the pigment gets deposited into the hair. Now, those that have a dominant allele are going to result in the pigment being deposited. Those that are recessive, so homozygous recessive individuals, will have no pigment deposited in their hair, so they'll end up being golden labs. So here we have all of these are black right here. So you can see black, and they have at least one of the dominant alleles for depositing the pigment into the hair, the coat. So they're all going to show their coat color, and then of course they'll show whatever color is dominant here. So these three brown labs are, of course, homozygous recessive for the color, or the color of the pigment, but since they show brown, right, so they must have at least one of the dominant alleles for depositing that actual color into the pigment. And then here, it doesn't matter what color is coded for the pigment. These are golden labs because they do not have a dominant allele for depositing for that gene that actually results in the depositing of the pigment into the hair. So if these did have at least one of those alleles, this one would be black, this one would be black, this one would be brown, and this one would be black. Sometimes the environment can have an impact on the phenotype. Now, the normal reaction is basically the phenotypic range, the result of a genotype that's influenced by the environment. So these characters are actually called multifactorial because they involve genetic components and environmental components. Some diseases in humans, which are considered multifactorial, are heart disease, diabetes, alcoholism, some psychological illnesses, and cancer, because they have both a genetic component and environmental influences. So, just to give you an example, you may be predisposed genetically to heart disease. However, if you live a lifestyle which is not conducive to developing heart disease, then you have that environmental influence, which, of course, in this case would be a positive. Let's look at these hydrangea flowers. So this is an example of environmental effects on the phenotype. So for these flowers, the color of the flowers, it depends on the pH of the soil. Now, both of these have the same genotype, but they have a different phenotype because of the pH differences in the soil. So here we can see if the soil is more acidic, then the flowers are going to be more blue. If the soil is more neutral, the flowers are going to be pink, and somewhere in between will be more purplish colors. Now, human traits often follow Mendelian inheritance patterns. Now, humans cannot be used ethically for genetic research for a number of reasons, right? So our generation time is relatively long. It's up to 15 to 20 years. That's a long time. You want to be able to use an organism that can mate and produce offspring relatively quickly, so you can actually do the studies. Parents in humans, really, we do not produce many offspring within a generation, if any at all. And then, of course, breeding experiments are unacceptable. We're not going to put some humans in a room and let them mate or tell them to mate and see what the outcome is. Again, it's not ethical, and uh, it would take too long. So these are studied by, or humans are studied by a pedigree analysis, which is basically a history of inheritance related to families that have mated. So it's a family tree essentially. A bit more about pedigrees. A pedigree is basically a family tree, like I said. It describes the interrelationships of parents and children across many generations. It's the history, right? So it shows the history of a particular family. Inherited patterns of certain traits can be traced through this history by using pedigrees to describe the family and their relationships. Pedigrees can also be used to make predictions about future offspring, knowing what happened in the past. And we could use the multiplication and addition rules to predict the probability of specific phenotypes as we try to make predictions about the future offspring. Let's look at a couple examples of pedigree. So here, I must kind of describe what a pedigree actually shows here. So we can see different generations, right? So um, at this first generation, well, as you can see, the males are going to be squares and the females are circles. Those that are affected, or those that have the trait that you're investigating, for this one, it's widow's peak. So what widow's peak is basically in your hairline, it comes to a peak near the forehead. Those with no widow's peak would be considered a continuous hairline. So we're studying that. So those that are affected are going to have the purple colored shading in their box or square. If individuals mated, then there's going to be a horizontal line connecting the box and square. And then a vertical line is going to connect to another horizontal line showing all of their offspring. Okay, so here we can see we have a male who has widow's peak, mated with a female who does not have widow's peak, and they had 4 children, right? So they had 3 males and 1 daughter. One of the males mated with another female, both of them having widow's peak, and then had 2 children, one of them having widow's peak and one of them not. And here we can see basically this is a relatively simple pedigree showing only 3 generations. Now you might be asked something like, is widow's peak dominant or recessive? In some cases, you might not be given the genotypes. You might be given other information where you have to determine the genotypes and then deduce the answer to a question like this. So looking at this, okay, widow's peak, is it dominant or recessive? Well, just looking at the genotypes here, you can tell that every individual that has at least one of the dominant alleles is affected. So you could say that this particular trait is dominant to no widow's peak, which it would be considered recessive. So those without widow's peak would have to have both the recessive alleles. Let's look at another. So here we're talking about the earlobes. There are what's called attached earlobes and free earlobes. So attached earlobes, the earlobe kind of blends in with the neck, and the free earlobes have like this hanging piece of ear that kind of hangs there. So again, we can look at those that are affected. We have three generations here, and then we have some matings. And looking at this, we have those that are shaded have the recessive genotype, right? So homozygous recessive, and this means that it's probably recessively inherited because those that are affected all have the recessive or homozygous recessive genotype, and none of the individuals with at least one of the dominant alleles has the free earlobe. Many genetic disorders are actually inherited in a recessive manner, but some of them are inherited in a dominant manner. So we're going to kind of talk about some of those. Now, the behavior of recessive alleles can occur, it really occurs differently depending on what other allele is present in that individual. So, recessively inherited disorders show up only when the individual is homozygous recessive, right? So, let's say it's a recessive allele. You must have both recessive alleles to actually show it. Now, those that are heterozygous, which they actually have one of the recessive alleles, which results in some sort of disease or disorder, are considered what's called carriers. So they're they're basically carrying that disorder, and this can be passed on to the next generation. Albinism is an example of a recessive condition, which is characterized by the lack of pigmentation in the skin and hair. So here we can see the possible genotypes. We have homozygous dominant, which is not albino, and then we have a carrier. This is the heterozygote. So these individuals are carrying the albino allele, or the allele that results in albinism. And then, of course, the homozygous recessive are going to actually show that particular trait. So they're going to. So here we can see this example. We have a cross between two normal carriers. So when you cross two normal carriers, you result in this particular distribution of possible genotypes. So here we have basically a 25% chance that their next child will be normal, and a 50% chance that their next child will be a carrier, and a 25% chance that their next child will be albino. Now, um, consang- consanguineous matings are matings between those individuals that are close relatives, or incestuous matings or marriages, right? So these types of matings are going to increase the chance of two carriers that are carrying a rare, let's say, recessive allele, which results in some sort of disease or disorder. So this is why you see an increase in particular disorders found in those types of matings within families. And it's because that those that are closely related are more likely going to have similar alleles, and they're more likely going to bring together two carriers of a particular, let's say, recessive allele. It will basically come out because of matings between these individuals. Now, let's go to dominantly inherited alleles. So some disorders are actually dominant alleles, and they can stay in the population or they could be weeded out of the population. So dominant alleles that are often lethal, okay, are often very rare. You don't see too much of that because if it's lethal early on in life where the gene doesn't get passed on to the next generation, then the individual dies before they're able to pass that or reproduce, right? But some diseases are late onset. like Huntington's disease. So, Huntington's disease is something that occurs later in life, and this is lethal. However, by the time the individual has reached the age to show the disease, they have already made it and passed those alleles on to the next generation. So they will be passed, and they can basically this particular disease will stay in the population. Achondroplasia is a actual form of dwarfism. This is caused by. So here we can see we have an individual who is dwarf, who mated with a normal individual. So the normal individual is going to have both the recessive alleles, of course. And again, we're talking about a dominant trait. So this dwarf individual has, in this case, one of the copies of the dwarfism allele. So when you show this mating right here, these two individuals have a 50% probability, or the probability is 0.5, that their next child will be dwarf, and 50% that the next child will have a normal height. Now, genetic testing and genetic counseling are usually done, actually done more often these days, right? In order to predict the probability of offspring or the next child or the unborn baby to have a particular disease. So genetic testing is the testing of the blood to identify any mutations or changes in protein levels. And then the counseling involves basically discussing and having this conversation, and the conversation and talking about probabilities and what it actually means. Okay, both of these kind of happen together. Now, a lot of diseases, probably more so now than I even know, have actual genetic tests where you could actually identify whether the offspring is going to have that disease or be a carrier. These days, more present day, we can actually determine that if you choose to. Now, fetal testing can be done in two different ways: amniocentesis and chorionic villus sampling, or CVS. Amniocentesis basically basically the taking of a liquid that in which the fetus is surrounded by, and that's tested. Chorionic villus sampling is basically a sample of the placenta is removed and actually tested. But I think these days now we could actually take the blood of the mother and actually determine a lot of the genetic abnormalities without being so invasive. Another technique would be ultrasound. Okay, so that's just kind of visualizing it. It's not as reliable as more advanced testing, well, these are more invasive, if you will. And then um, blood testing is probably the way that we're moving towards these days. Now, just looking at these, you can see that the amniocentesis, basically they have this long needle which punctures through the placenta to obtain some of the fluid in which the baby is swimming in. And then the chorionic villus sampling is not as invasive as the amniocentesis. However, it is somewhat invasive, and it requires taking a sample of the placenta. Both of these have their drawbacks and associated things that could go wrong, but they are quite useful. So what would happen is basically this is a sample, and then the genetic and biochemical test would investigate maybe protein levels or enzyme levels, and then of course a karyotype would be obtained from this as well. These types of testing, you can get the karyotype, determine whether there are mutations in the number of chromosomes, mutations in the chromosomes in general, and you could tell whether the child is male or female. All right, so in summary, this segment talked more about the non-Mendelian inheritance, those types of factors. We did discuss a little bit about pedigrees. I will show you an example in another segment. and genetic testing.